Okay, I want to continue on talking to the second half of this Monday lecture, talking about DNA, the structure of DNA, the function of DNA, and so you've got this this structure of DNA with the nucleotides, and what's it used for? What does that mean? Why are we spending all this time talking about the structure of DNA? Because the structure is fundamental to the function. So I left off with the last lecture talking about the double helix with these paired bases. And I wanted to make the point that, again, you see, here's somebody else's representation of these. You've got a pentose sugar, those pentagon-shaped sugars that are joined with a phosphate, again, a P that's yellow and a circle as related to with energetics and the forming of those bonds between the nucleotides. You've got a bunch of nucleotides, sugar, phosphate base with a T, sugar, phosphate base with a C, sugar, phosphate base with a C, sugar, phosphate base with an A. So these are all nucleotides along one half of the strand of DNA. Here's the complementary nucleotides. The bases are paired up according to that. Every A is going to have a T. Every G is going to have a C. AT is all that you see. Every A was with a T. Every G is with a C. There's only 4 of these, so it's not really that complex, which is one of the reasons why it's amazing what this DNA structure does. Another thing that you notice with these base pairs is that they're bound together by bonds. They're hydrogen bonds. So these little dashes you see between the A and the T, there's 2 of those. Dashes you see between the G and the C, there's 3 of those. There's either a double bond or a triple bond that's formed between a complementary base pair, these nitrogenous bases. This is the middle of DNA. This is the middle of the rungs of that ladder. It has these bonds in it. And that is relatively weak. These bonds with little dashes are weaker than the bonds between the base and the sugar and the base and the phosphates and the phosphates and the next sugar. These bonds, they can be broken, but they tend to be stronger than these bonds between the bases. This is the weakest part of DNA. So we're going to see when we look at DNA being copied and when we look at proteins being made from DNA that this DNA structure is going to be broken apart. Where is it going to be broken apart? In the weakest part. The weakest bonds are going to be broken, and these are the weakest bonds. So when we look at DNA being copied, replicated prior to cell division, this is where the bonds of DNA are going to be broken, right here. When we look at DNA being used to make a protein, first of all by synthesizing RNA, where do you think the bonds in DNA are going to be broken? Well, you're probably all saying right here. And that's right. This is where those bonds are being broken. Okay, well, let's look at this first phase. We're going to look at 2 different things happening with DNA. First, we're going to look at DNA being used to make more DNA. You know that happens because we spent all this time looking at mitosis and meiosis and cell division, and we saw all these chromosomes being copied, and you realize that we all started out as a single cell, and now we're billions of cells. We've gone through billions of cells. So literally, billions of times, our DNA has been copied, our chromosomes have been duplicated, and passed on to new cells. So this happens all the time. And in most of us, there haven't been mistakes that have been made. We don't have mutations that have taken place. Maybe there's some people certainly where mutations have occurred. But this copying of DNA and copying of chromosomes happens with an amazing amount of accuracy. So DNA has all this information that new cells need. And if you think about it, the DNA, it's got the sugars and the phosphates, those are all the same. Those bases, there's only 4. But the sequence of bases, which base is first? Is it a G, or is it a C, or is it a T, or an A? One of those 4 is first when you're going down the chain of nucleotides. And then one of them is second, one of them is third, and another one. There's a certain order of those bases. The sugars are the same. The phosphates are the same. But the order of bases is different in me and you. It's different in us, in humans, and in some kind of other animal or a plant. So if you can take the sequence of bases in DNA, that it's a G and an A and a C and then a T and a T and another A and an A and a T, that specific sequence, if you can copy that sequence and use it to make more DNA, then that is a template. It's a blueprint. You don't just take any nucleotide. You take a nucleotide with a G first. And you just don't take any nucleotide. You take a nucleotide with an A second, whatever the order happens to be. That's the key for copying DNA without making mistakes. That's the key for making DNA that's the same as the original DNA. So that's what we're going to look at, not in any great detail. You know, there's these 2 strands of DNA are actually being copied differently in different directions. It just adds to the complexity of trying to understand this. And we don't need to get into that and look at these fragments that are being formed and the 5' end and 3' end. All we're being concerned with is these bases being a blueprint, the sequence of those bases, or the sequence of those nucleotides, however you want to look at it. Each nucleotide is a base. The sequence of those is the key to making more DNA. We'll see later it's also the key to making proteins. So here you've got a DNA molecule. You recognize it here, and again, this artist has put these purple ribbons as the sugars and the phosphates, and you see those bases in the middle joined together. And there's bonds between the T and the A and the G and the C. There's bonds between each one of those. Those bonds are broken as this enzyme moves along the DNA molecule and splits it apart. It splits it apart between the bases. There's an enzyme that does that. It splits the bases apart and it exposes a base that just a second ago it had a pair. There was an A that was paired with a T. Now an enzyme comes along and breaks a bond between the A and the T. And now this A is sticking out there, flapping in the breeze. Look, it doesn't have a pair. It's not bound to any other base. It's looking for a base. This is an A. The only base that this A is interested in is a T. There's also a bunch of nucleotides, individual nucleotides, a sugar and a phosphate and a base. There's a bunch of nucleotides where the bases are a T, and there's some that have a G, and there's some that have an A, and there's some that have a C. There's all those nucleotides with all 4 bases. Now this A that's sticking out there, it doesn't want just any base, it's not happy with any base. It wants a T. It's an A, it wants to form a bond with a T. So there's another enzyme that comes along and can take a nucleotide, just a nucleotide, one sugar and a phosphate and base, and in the case of a base that's a T, this enzyme will facilitate the joining of that A that's lost its complementary base when that other enzyme came along and unzipped DNA. It'll take the nucleotide with a T and join that with the A. And it'll also join the nucleotide with the rest of the DNA chain. So that's what we're going to see when we look at what happens with DNA synthesis. So as I said, here's this enzyme that comes along and splits DNA. Here's a DNA molecule you see here. This enzyme comes along and breaks the bonds between the bases. So a minute ago you had this yellow A and this green T, and now you've got an A, you've got this A and another A, you've got 4 or 5 A's out here, none of which have a complementary base. Just a minute ago they had a green G if they were a C, or they had a green T if they were an A. So here comes this other enzyme. It's taking this little thing right here is a sugar and a phosphate, and this green part is a base. This base is a complementary base that goes along with the DNA, the other base. Here's what's happening here. Another way of looking at this, well, there's 3 different depictions of what's going on here. You've got, it's not shown here, but there's an enzyme that's coming along and breaking these bonds. This shows the letters, at least. It shows the bases. So here's an A and a T that were joined together. There was a T that was with an A. Now in this case, here's this nucleotide all by itself, this A, that's attached to a sugar and a phosphate. There's this enzyme, this yellow enzyme that's moving along here, that's taking this nucleotide with the A and forming a bond with the T. It's facilitating that. So as this moves along, it's got all this blue nucleotides, all these G's and C's and A's that are attached to sugars and phosphates. Just a minute ago, those were part of the original DNA. But you've got this original part and the blue going up this way, and you've got the original part, the other part of the original DNA going this way, and now you've got red DNA that's new, that's being formed when these new bonds are being formed. You've got one little nucleotide that's out there all by itself with a sugar and a phosphate and a base. That base is an A, so it's looking for a T, and now you've got a new strand of DNA that's half old, or original, the blue, and half new, that's red. So in this way, DNA replication is referred to as semi-conservative, where half of it is old and half of it is new. In the end, if you look at this, you've got this strand of DNA here, and you've got this strand of DNA here. These are being formed differently, fragments. But again, we don't need to be all concerned about that. But you've got this strand of DNA and this strand of DNA, half new and half old, but they're identical. They are the same DNA as this original one. But you don't just have one. This is one strand of DNA to begin with. When you're done with this, you've got 2. One up here and one down here. This one is half new and half old. This is half new and half old. So there's another way of looking at it. There's another picture of this coming along and breaking the bonds between the bases. And again, here you've got these little deoxyribonucleotides coming in one at a time that are being joined to make a new strand of DNA, half old, the blue, and half new, the red, and another enzyme that's doing this in fragments. Here, this one, and then that one, and this one, and that one. But this is going this way, and this is going that way. That's the direction for the, or that's the reason for the 3 and 5 prime ends. But again, we're not going to get all caught up in that. So we start out with DNA. The goal is to have 2 sets of DNA, 2 sets of genes, 2 sets of chromosomes. This is how that's done. You've got 2 different strands of DNA that are the same. They're identical. And then those can be passed on to 2 new cells when a cell divides. Well, here's another way of looking at this. Yet another way. You've got this enzyme that's moving along, splitting the DNA, exposing those bases, and you've got another enzyme that's coming along and facilitating the formation of this is one new strand and that's another new strand. They happen different ways. But you started out with this original DNA, split in half, and you end up with one segment of DNA and another segment of DNA that are identical to the original. That's how you copy DNA, and that's how you can pass new chromosomes onto new cells. The instructions for doing that are very specific. If you think about, go back to this slide, the base, that's the order of these bases is conserved. Those aren't changed. This is the order of bases that are in somebody's DNA. That's the order of bases that are in the new DNA that's made and the other strand of DNA that's made. They're exactly the same. So these types of figures, they just show the general process taking place. But if you looked very specifically and you saw the order of bases, which is the key to what makes me different from you and you different from everybody else, is the order of those bases. That's preserved because if you've got an A, the only base it's going to form a complementary pair with is going to be a T. If you've got a C that's supposed to be the 357th nucleotide is a C in the original DNA, then the 357th nucleotide in the new DNA, both strands of the new DNA are also going to have a C at nucleotide number 357. And it goes on like that for billions of these base pairs, like I said. Okay, so what we're going to look at is this idea that you can take DNA that's in a chromosome that contains all these genes, and you can copy this DNA, and you can make exact duplicates of the original DNA because of the order of those bases and the way that these bases pair with the complementary base pair. Not just any base, but complementary base pair. And so you can make new DNA that's exactly the same as the original DNA by following the rules of which base pairs with which other base. And now the next step is for us to see, okay, well, you can make new DNA. That DNA, even when you copy it and make new DNA, is sitting in the nucleus of a cell. It's not doing anything. So how do you use this DNA to make me or you or whatever organism you're talking about, a whale? That's what we're going to look at next. And we're going to see the same thing, that the order of bases in DNA is specific to an individual. That's how you can tell one individual from another. You can do a DNA test, and there's parts of DNA that differ amongst individuals. The difference is not DNA in general, it's the sequence of those bases in DNA. It's the order of the bases. That's what allows you to tell different people apart. That's what a DNA test is.